Hi students, as usual we are again back with another previous year question from NEET biology. Welcome to UNS classes. Let's directly go to the question. Here today the question is about histone. Let's see what the question is. Which one of the following statement about histone is wrong? So directly before going to the option, let's learn a little about what is histone is. So what is histone? Histone is nothing but protein which helps to wrap the DNA around it to make the DNA compact. So how much DNA is tightly wrapped on the histone depends upon the different stages of the cell. Okay, when there is cell division taking place, DNA will be compactly packed around the histones to form chromosomes. So let's learn little more about histone. So what is histone? Histone is nothing but a protein, right? Histones are proteins responsible for DNA packaging, right? Now, then histones are, see, positively charged. They are positively charged proteins. If histones are protein, if the histones are positively charged, what will be the charge of the DNA? Obviously, it will be negative. So, it helps them to tightly bind each other. See, hence, the DNA is negatively charged, hence they can easily bind to each other. And what are the majority of the amino acids present in the protein called histone? They are mainly with arginine and lysine. See, when we say arginine and lysine, when we speak about amino acid, there are acidic amino acid, aromatic amino acid, basic amino acid. Here arginine and lysine are basic amino acids. They are basic amino acids. So arginine and lysine are the majority of the amino acids present in the protein called histone, and arginine and lysine are basic in nature, right? Now, next, histones are involved in controlling the gene expression. That's true, right? Because I told you the histone helps in DNA packaging. If we have to transcribe or translate an information from a gene, this DNA should not be tightly packed around the histone. So whenever in the body had to express a gene, this DNA wound to the histone will be loosely packed so that the information present in this DNA can be transcribed into an mRNA. From mRNA, protein is made. Whenever in the body does not want to express a gene, at that time the DNA will be tightly bound to the histone protein. So the DNA won't be available for transcription and thereby translation. So it is true that they control the gene expression, right? Now, there are different types of histone proteins. Different types of histone protein means H1, H2A, H2B, H3 and H4. This whole histone proteins have been divided into two types. They are core histone and linker histone. So what is core histone and linker histone? Core histone is nothing but, see, the core histones are H2A, H2B, H3 and H4. These four histones comprises to form the core, and this H2A, H2B combine to form a dimer. What? Dimer. Or we can write it here. See, H2A and H2B combine to form dimer. What kind of dimer? See, both of the proteins are different. H2A is different, H2B is different. So we can call it as heterodimers. Okay, so they form heterodimers, right? So H2A, H2B combine to form heterodimer. Similarly, this H3, H4 combines to form heterodimer. Here H3 and H4 combines to form what? Heterodimer. Okay. So, and the next thing is that in the core there will be two copies of H2A, H2B heterodimer. That is H2A, H2B 2 times. So there will be 2 copies of H2A and 2 copies of H2B. So in total 4 histone protein will be there in H2A, H2B 2 times. Similarly, H3, H4 heterodimers will also have 2 copies of it. So in total here also 4 protein will be there, right? So if both of this combine, how much is total? Total is 8. So the core is also called the octamer. Octamer means 8. See here the core histone H2A, H2B, H3 and H4 forms an octamer. See, they forms an octamer. Now let's see the only histone which left is H1 here, right? So let's see what H1 does. H1 is also called as linker histone. Linker histone means it locks the DNA in place when the DNA is properly wound around the octamer, right? So if you see this image, this is and this blue color ball hole is the octamer and this white is the DNA. Okay, so when the DNA is properly wound around the octamer means what and all will come here. This is H2A, H2B heterodimer. How many heterodimers of H2, H2 will be there? 2 copies, right? So this is also H2A, H2B, right? Now which is the another heterodimer which is left is H3 and H4. This is H3, H4 heterodimer and this is H3, H4 heterodimer. Totally how much is formed? 8 histones and the octamer is formed, and around the octamer the DNA is wound properly, and once the DNA is wound properly it is kept in place by an linker histone. That linker histone here is H1, right? Now all basic things about histone we have learned. What we have learned? Histone is nothing but a protein around which DNA is wrapped or DNA is packed. What is the charge of the histone? The charge of the histone is positive charge and DNA is negatively charged, right? Now majority of the amino acids present in the histone proteins are arginine and lysine. Okay, and what is arginine and lysine? Is an acidic amino acid or a basic amino acid? They are basic in nature, right? And now what are the different types of histones involved in this? They are H1, H2A, H2B, H3 and H4. These H2A, H2B, H3 and H4 forms the core. They are called the core histones and they are totally octamer. They are 8 in number, and the last one which is left is H1. H1 is also called linker histone. Okay, now let's go back to the question. Here, what is the question? Which one of the following sentence is wrong about histone, right? So let's see the first option. In the first option they are saying that histone carry positive charge. Is it true? Does histone carry positive charge or not? Yes, right, because the charge of a DNA is negative and the charge of a histone is positive. So this sentence is true. Now the second sentence are histones are organized to form a unit of 8 molecule. Is it correct? Octamer, right? Octamer means how much is 8. So 8 molecule is there. So this sentence is also correct. Now the third one is the pH of histone is slightly acidic. Is it correct? The majority of the amino acids present in the histones are arginine and lysine, right? Arginine and lysine. What is the one letter from arginine and lysine? We can write arginine is R and lysine is K. Both are they acidic or basic? They are basic in nature, right? Basic in nature, right? So is this sentence correct? The pH of the histone is slightly acidic? No, right? The pH of histone is basic or alkaline in nature, right? And the last option is histones are rich in amino acids called lysine and arginine. Is it true? See, lysine and arginine. So this sentence is also true, right? So which is the correct option here? They are asking which sentence is wrong here. Here which sentence is wrong? Option C. So here the answer is option C, that is the pH of an histone is normally alkaline or basic, not acidic. Hope you understood about histone and got the answer. Thank you for watching. Happy learning.